Hello ladies and gentlemen, welcome back to C.A.R. Entertainment. I am your host Dylan Hamilton, and today we're going to talk about gene transfer as we continue on with our gene technology topic in A22 biology, guys. We've talked about PCR and genetic fingerprinting, and now we're going to move on to gene transfer. So for further ado, let's get into it. So, definitions. You know quite a lot of definitions for gene transfer. The only real main one is gene technology, which I've actually already told you about. But gene technology, just a reminder, is the manipulation of genes for the benefit of people in agriculture, food production, and medicine. The other ones, basically, if you just know sort of what they are, you probably will be asked a definition on them. If you know what they are, it'll be greatly beneficial to yourself. So we'll go through and talk about them. Right. Restriction enzymes are enzymes that cut DNA at specific points, make the small fragments. DNA ligase will join DNA fragments together. Vectors will carry DNA into cells. Plasmids are commonly used vectors. Genetic markers identify cells that are going to be transformed. PCR amplifies small sections of DNA. cDNA makes a copy of DNA by a copy of RNA. It makes a DNA copy of RNA. DNA probes identify and label a piece of DNA which contains a certain sequence, and DNA sequences is whenever you read a base sequence of a length of DNA. So we'll move on to GEMs, which are genetically engineered microorganisms. And how you do this is that you obtain a required gene from a bacteria or whatever. You insert the gene into a vector. You then insert this vector into a cell. You identify the host cell that then took up this gene, and you clone that gene that is modified by the host cell, and so you get the desired product, and you'll extract it and purify it. Let's go through how this works, but first we're going to talk you through DNA processes because these are used in this process. You will identify a section of DNA which contains a specific sequence of bases using restriction endonucleases to cut out a desired section. The DNA probe will be labeled using a radioactive or fluorescent tag, and the DNA section that is cut out by the restriction endonucleases will be separated by gel electrophoresis and transferred to a nylon membrane. The then fluorescently labeled DNA probe will be added to target a sequence that is present where the DNA probe will hybridize. The nylon membrane will be exposed to UV light, and the DNA probe will appear as a fluorescent band. This, as you know, is known as genetic fingerprinting. So the first note is that you wash off the DNA probes that do not attach, whether or not they show up hybridized or not. Note 2 is that the radioactively labeled probe that is used and the nylon membrane are exposed to X-ray film. The probes targets the sequence combined with the fogging of X-ray film. So gene transfer 1, how does it work? Right. Well, gene transfer, how do we obtain the required gene? We're just going to go through all of these sections here. So how do we obtain the required gene? Well, restriction endonucleases will cut DNA double strand by hydrolysis at the specific sequence of bases called the recognition site. They will sometimes cut straight across, leaving two strands of a cut section with blunt ends, but more often they make staggered cuts, so they leave sticky ends. And the advantage of these sticky ends is that they're able to join ends with another section of DNA. You can also obtain the required gene by using the enzyme reverse transcriptase. You would do this by isolating and extracting mRNA of a desired gene. You would then use this to make a strand of DNA, also known as copy DNA, using this RM and mRNA as a template following the base pairing rules. The enzyme DNA polymerase will make a double-stranded DNA from the strand of copy DNA. So now that we've got the gene, we have to put it into a vector. The main types of vectors you will use are plasmids and viruses. So how do we do this? Well, you'll get a bacterial plasmid and a closed loop of DNA. You will then cut this to create ends of which the donor DNA will join to. The same restriction endonuclease that is used to cut the plasmid is used to exercise the donor DNA. And this is because then the plasmid and the donor DNA can make a perfect join, and the bases will be paired up so DNA ligase can enable the DNA backbone through phosphodiester bonds. The plasmid DNA is now called recombinant DNA, and the plasmid is referred to as a recombinant plasmid. In terms of inserting the gene into the host cell, you get the bacterial cell and incubate it with calcium ions and subject it to heat shock, and therefore the cell is more likely to take up the recombinant plasmid. The treatment will make the bacteria more permeable, and therefore it reduces the barrier to taking up a small proportion and a small proportion of bacterial cells will actually take up the recombinant plasmid. So how do we then identify the host cells that have taken up the gene? Well, we'll use marker genes by having a bacterial plasmid that is almost always containing genes that are included in the development of antibiotic resistance in bacteria. Some confer resistance to, such as the R plasmid, which is genes for resistance to antibiotics tetracycline and ampicillin. If you cut the R plasmid prior to inserting the donor DNA, the destruction enzyme will cut the bacterial plasmid from one of these genes, and this will provide antibiotic resistance. The donor DNA will then annihilate effectively into the plasmid, which will no longer have the gene to confer resistance to tetracycline, and ampicillin will be unaffected. There's a wee diagram up the top to take you through that. You get your host DNA. It is then put into agar that contains tetracycline. These tetracycline then is moved in to one where you have recombinant plasmid that contains tetracycline, and then you have an agar that contains ampicillin and tetracycline. So eventually you then get the point where you have colonies of recombinant plasmid and an agar that contains tetracycline, and with agar that contains ampicillin and tetracycline. So at the very end, essentially, you will then see which dots are not removed. So all that black arrows there and those last ones, you see those are the ones that were not removed. So they are operating in both tetracycline and the ampicillin and tetracycline. So those are, sorry, it's the ones that have been removed, sorry, I should say. So the ones that have been removed are the ones that offer resistance. So using fluorescent marker genes and gene transfer as well. So using fluorescent marker genes, you know organisms will possess genes that produce fluorescent proteins. These genes will be isolated and attached to the gene of interest, and they're marked since they produce a fluorescent protein. These attached genes are then inserted into a vector and carried into the bacterial cell, where the takeout will be readily identified and the transformed bacteria will show under UV light. The bacteria will be identified and transferred to a sterile nutrient medium and increase in number. Cloning the gene in the modified host cell, they will be grown in large fermenters, and the desired product will be extracted and purified. Thanks for watching the video. I've been Don Hamilton from C.A.R. Entertainment, and I'll see you next time.